Hi guys. Before moving to the topic, let's observe this picture. This is the latest technique they used to diagnose the particular brain damage. So this is the caloric stimulation method. They used to find if any damage occur in the brain or not. They used to take this procedure and they used to find this is the latest technology, caloric stimulation. So let's move quickly to the topic. So today we are going to discuss about the virus. So viruses are the smallest known infective agents which may cause the disease. So these are viruses are smaller than bacteria. Viruses do not possess any cellular organizations and they do not fall strictly into the category of unicellular microorganisms. They are acellular microorganisms. Viruses having many shapes and different sizes depends upon the viral families. Okay, they're classified into filamentous, isometric, enveloped, and non-enveloped head and tail. Four groups of viruses. So filamentous viruses are long and cylindrical. Let's see the properties of viruses. So they are acellular microorganisms. They used to carry only one nucleic acid, either DNA or RNA. In some specific RNA, they used to carry RNA. In some specific viruses, they used to carry only DNA. They are obligate intracellular parasites. Viruses are not affected by antibacterial antibiotics. Okay, they multiply by a complex processes and not by a binary fusion. The binary fusion processes they used to undergo in bacteria. Unicellular bacteria undergo binary fusion reproduction, but not viruses. They multiply differently, and within a second they used to multiply the viral components. And let's see the differences between the virus and bacteria. So bacteria we can observe in microscope, normal compound microscope we can observe the movement of bacteria. But the motility of virus we can observe in under this ultra microscopic. So viruses is acellular, bacteria is unicellular, as we know. So some examples of viruses are measles virus, pox, and influenza viruses, AIDS viruses. Okay, bacteria we have discussed about bacteria. Some bacteria is Escherichia coli, tetanus bacteria, pneumonia bacteria, cholera bacteria. So the main difference of the virus and bacteria is virus is even smaller than the bacteria. Virus is acellular, bacteria is unicellular. Let's see the structure of the virus. Okay, extracellular infectious viral particle is known as virion. The scientific word virion is nothing but the extracellular infectious viral particles. Let's see the size range of viruses, 20 to 300 nanometers. The smallest virus till now they have observed is parvovirus. The size of parvovirus is 20 nanometers. So the biggest virus they have observed till now is pox virus. The size of pox virus is only 300 nanometers. See how tiny they are, right? We can't observe this virus in the light microscope. We need ultra microscope to observe the virus. So measuring the size of the virus, we are having some apparatus or instruments to measure the size of virus. If you see in microbiology laboratories, we can observe all these apparatus or the instruments for measuring the size of the virus. Shape of the virus. So we have discussed that depending upon the family of virus, they are different in shapes. So for example, pox virus are in brick shaped. So rabies virus are in bullet shaped. So tobacco mosaic virus is in rod shape. So like bacteria, virus is also having different shapes. It depends upon the viral families. So generally all animal viruses are roughly in spherical, irregular and pleomorphic shapes. Okay. See, the bacteria, this Escherichia coli, is 3000 to 1000 nanometers in length and breadth. If you see the Ebola virus is 970 nanometers. Here, tobacco 250 nanometers, rhinovirus 30 nanometers. Okay, let's see the structure and chemical composition of the virus. So, viral capsid. The virus consists of nucleic acid core surrounded by a protein called a capsid. Here, in this picture, you can observe to the proteins, they are surrounded by this particular protein called capsid. The DNA or RNA, for example, the chromosome is protected by a protein called capsid. Okay, capsid. This particular capsid is composed of a large number of capsomeres, which is made up of polypeptide molecules. Okay, this particular capsid is made up of polypeptide molecules. The capsid with enclosed nucleic acid is known as nucleocapsid. So here this is a nucleocapsid. So what are the functions of capsid? It protects the viral genome from physical destruction and enzymatic inactivation by nucleases in the biological material. So, how we are protected? Our every cell is protected by a cell membrane like that. Here, for virus, their contents, the viral contents, components are protected by this capsid. Okay, and it provides a binding site which enable the virus to attach the specific site on the host cell. So specifically this nucleocapsid is responsible for attaching to the host cell. For example, if any virus is want to enter, has entered in your body, means for attaching purpose, for host cell means our cell. So then for attaching purpose it is required, the capsid is required, and it will release the viral content for making complications in your body. And it facilitates the assembly and packaging of viral genetic information. While replicating this virus in your body, this capsid is forming or making the assembly and packaging. It will package the viral genetic information into one part. It is nothing but the capsid. And it also serves as a vehicle of transmission from host to another cell. So after replicating lots of viruses from movement of one cell to another cell, this particular capsid is responsible for the movement of a transmission, the vehicle transmission, acts as a vehicle transition, and it is also act as antigenic and specific for each viruses. Okay, it provides a structural symmetry to the virus particle. It's also responsible for the structure. Okay, then that is about the capsid. Then we'll discuss about the viral symmetry. So this is nothing but is grouped in three types based on arrangement of morphological subunits. So one is icosahedral symmetry, another is helical symmetry, another one is complex symmetry. So viral symmetries. So we'll see one by one. Icosahedral symmetry. Icosa means in the Greek word it is 20. The particular virus is made up of icosahedral symmetry shape. Okay. Into the corners it brings the shape of 20 facets or sides. Each facet is in the shape of equilateral triangle. You can observe this picture, equilateral triangle, like the 20 facets they will observe this particular bacteria virus. Okay, icosahedral type of virus is adenovirus. If you see the structure of adenovirus, you can observe like this. And helical symmetry. The nucleic acid and capsomers are wound together in the form of helix or spiral. So that's why they kept the name helix symmetry. In helix shape or the spiral shape, you can observe this particular virus like as influenza virus, rabies virus, para-influenza virus. If you see the complex symmetry, the virus which do not show either icosahedral or helical symmetry, it is in complexity form. It makes some complex symmetry, it makes some complex. For example, pox virus. So depending upon these viral symmetry, this particular virus structure may observe. Okay. So that is about the viral symmetry. Viral envelope. The virions may be enveloped and non-enveloped. So enveloped viruses are there and non-enveloped viruses are there. So enveloped viruses means it is the virus contents are covered by a lipid like a plasma membrane. So however cell organelles are covered by a lipid layer, cell membrane like that, the viral contents is also covered by a plasma membrane. It is made by the lipids and is also responsible for the attaching to the host cell and releasing the viral contents for making damage. So this particular covering layer, the envelope, is a glycoprotein in nature. These enveloped viruses are susceptible to the action of lipid solvents such as ether, chloroform, and detergent. So enveloped viruses are herpes simplex virus, hepatitis B virus, and HIV virus. So non-enveloped virus. So viruses which does not have any outer covering. They are the viral contents and come into the particular cytoplasm, make the replication and move out from the host cell by damaging the host cell. And it will damage again, it will move to the another host cell and make the damage. Like that, it will damage all the cells which is present in your body. So that is about the enveloped and non-enveloped virus. So peplomers. In mature virus particles, the glycoproteins are often appearing as a projecting spikes. On the surface of the specific virus, you can observe the projecting spikes. That is nothing but the peplovirus. That is the outer surface of the envelope. Peplomers. So present pandemic, the coronavirus is there. The present coronavirus having the projecting spikes on the surface, so that the spikes can attach to the host cell and can easily enter into the human cells and making this particular damages or complications. So peplomers. The virus have more than one type of peplomers. For example, influenza virus carries two types of peplomers, the hemagglutinin and neuraminidase. The functions of peplomers: it helps to attach the host cell for the entrance into the particular cell. It attaches the particular receptor, for example, on the red blood cells, causing these cells to agglutinate. And it has also enzymatic activity like neuraminidase, which cleave neuraminic acid from the host cell glycoproteins. And it also has antigenic properties. Peplomers, very important. See, here the peplomer, the spike shapes, the end spikes you are observing, right? End spikes, head spikes. So, the viral nucleic acids. The virus contains a single kind of nucleic acid, either DNA or RNA. what we have discussed, which encodes the genetic information for the division, the replication process. Specific virus, they will carry the DNA only. The specific virus, they will carry only RNA for the replication purpose. Okay, either single stranded or double stranded. So according to the nucleic acid present, the virus can be classified into DNA viruses and RNA viruses. So for example, in some books you will find that DNA viruses, RNA viruses, like this. Okay, see, DNA viruses and RNA viruses, these are specific examples. So how viral will multiply or how virus will replicate, we'll see the process of how, what are the steps undergo for the replication of particular virus. See, first is attachment or adaptation and penetration. The next step is uncoating, and the next step is biosynthesis, next maturation and release from the host cell. We'll see one by one. So adsorption or attachment, it is mediated by binding of virion surface structure of, for example, ligands. How the ligands will bind to the specific receptors which is present on the cell surface. Like that, this particular virus will, the help of their structure, which will attach to the specific receptors or enzymes to enter inside of the cell and making the damage, making the changes in the particular cell. So for example, in case of influenza virus, they're having the surface glycoproteins they're having, it will bind to the specifically to celiac acid residue of glycoprotein receptors and making complication to the respiratory system. For example, HIV surface having glycoprotein 120, it acts as a ligand, binds to the CD4 cells, cell differentiation 4 type cells of T lymphocytes. T lymphocytes are responsible for the immune response. So this particular virus is deactivating the immune response by binding to the specific receptors. So like that, it will damage the particular our body or making the complication of the specific systems. Each and every specific virus, it will make to the make complications specifically in your body. So human immunodeficiency virus, right, HIV. So this particular virus, it will act on the immune response cells or the particular receptors for deactivating the immune response. And what will happen if you deactivate the immune response will not happen, means all the microorganisms will easily enter in your body and make some complications. Okay, attachment, first step is attachment. First they need one host cell to attach, then after attachment what they will do, see, there is a particular virus attaching to the particular receptor, okay, with the help of the spikes proteins, the glycoproteins, like this. Okay, you're observing, right, the virus is coming and binding to this particular receptor. Then what will happen? Penetration, it will enter. After binding to the virus, particle is taken up inside the cell. Okay, mostly non-enveloped viruses enter by receptors mediated endocytosis. Only the enveloped viruses, they need some specific fuse their membranes with cellular membranes to deliver the nucleocapsid or genome directly into the cytoplasm. Okay, enveloped and non-enveloped viruses. See, after entering this, they are releasing their viral contents into the cytoplasm and making the complications. So this is one of the RNA viruses. They are releasing the RNA genetic information into the cytoplasm. Okay, this is about the penetration. Then uncoating. What means uncoating? What it is uncoating means, it is a process of stripping the virus. They are releasing the nucleic acid. What you have seen in this observing, they are uncoating. That means they are moving the dress. See, this is the uncoating process. They are releasing their genetic information into the cytoplasm. That is nothing but the uncoating released into the cell. Most of the viruses uncoating is affected by the action of lysosomal enzymes of the host cell. So, in ourselves, we are having lysosome, they are having digestive enzymes. Whenever this particular microorganism has entered in your body, means they'll make into the pieces, they will protect us. Okay. Then what will happen after penetration? They will make some uncoating, release of genetic information. After that, replication starts, biosynthesis, synthesis of viral particles with the help of some enzymes and proteins. They will utilize our own enzymes and proteins and making some mutation to the particular proteins and enzymes for the synthesis of viral contents. Okay. So in addition, regulated proteins are also synthesized, which are served to shut down the normal cellular metabolism. So for, for viral replication, they will do after this particular uncoating. They used to synthesize the viral content in your body, and also additionally they used to produce the regulatory proteins. That particular proteins will stop the activation normal cellular metabolism process, and it leads to sequential production of viral components inside the specific cell. Okay, that is a nothing but the biosynthesis. Here the synthesis of lots of virus is produced in the particular cell. After that, it has to move out from the particular cell. While moving, some viruses, while move out from the particular cell, it will kill the particular host cell. Either it will damage the particular cell. Whenever they need, they used to produce continuously viral components. Okay, see here, they are making some viral synthesis. See, virus. After that, it will move out from this particular cell. Either damage of particular this cell, either damaging, and lots of viruses will move out from the cell. Okay, see once again. So new virus is creating here with the help of some regulatory proteins. Lots of viruses will produce, then move out from the cell, and these lots of viruses will move out to each and every cell. Only one virus is entered in the particular cell and making the replication of particular viruses, like lots of viruses, for example 50 viruses they have formed. It will move out from the cell, and that 50 viruses each will enter into the each cell, and again 50-50. See, like that it will damage our cells. So steps of biosynthesis. So transcription of messenger RNA, obviously messenger RNA for viral nucleic acid, then translation process will occur, then protein synthesis will happen, then viral component has produced the replication of viral nucleic acids. It will produce, make some daughter virus virions and everything. Daughter viruses will produce, and it will move out. After synthesis, it will move out from the, before moving out from the particular cell, it will get matured. Assembly of viruses, various viral components, it will make some maturation. So what you have seen in that last peak, after getting maturation, that means initially it was in growing stage. After getting matured only, it will make the complication to the other cells. It may takes place in either in the nucleus or in the cytoplasm. Some viruses they make replication in the cytoplasm. Some viruses they make replication in the nucleus. That depends upon the enveloped virus and non-enveloped virus. So, in case of enveloped viruses, the envelope are derived from the host cell, nuclear membrane, plasma membrane. Then the release, moving out those viruses, the virus daughter cells from host cell to outside. The viruses can be released from cells after lysis of cell. I told right, either damaging either lysis of the cell or maybe damage or budding from the cell from the plasma membrane through exocytosis process. That process is nothing but the exocytosis. So viruses that exit naked nucleocapsid may be released by the lysis of the host cell or reverse phagocytosis. Okay, the release of enveloped viruses occur after budding from the plasma membrane without killing the host cell. Some specific viruses they won't kill the host cell because they want to produce lots of viruses. For that purpose, they won't kill the host cell, they will make changes in the particular cell. So only the virus components will replicate in the particular cell. They will damage our normal cellular metabolism. See here, this is about the enveloped DNA virus. Here, this is the non-enveloped RNA virus. So, envelope they will bind into the particular receptors. It will engulf. See, it will enter and make attachment here. Then making the penetration. Then uncoating. Then after that, those viral DNA material or genetic information has moved into the cytoplasm. It will make into the nucleus, enter into the nucleus, and make the changes in the expression and make the replication of particular RNA, and will move out from the nucleus and make assembly or maturation and release finally. Here, non-enveloped RNA virus directly crosses the cytoplasm, this particular cell membrane, and making some penetration, uncoating, replication in the cytoplasm, making some maturation and move out from the nucleus. Okay, so this is about the overview of virus reproduction. So particular virus is binding to the specific receptors, attachment, penetration, uncoating, making the transcription, genome replications, translation process, and making and maturation assembly, then release from the particular cell. Like this, the virus will enter in your body. So this is about virus. Thank you. Have a nice time.